Hey, thanks everybody for coming. And thanks to the organizers, Jasmine and Dominic, for inviting me. So I'll start off by just positioning us a little bit. We're here today in Toronto on the traditional land of the Huron-Wendat, Anishinaabe, Haudenosaunee, and the Mississaugas of the Credit. Today, Toronto is home to many Indigenous people from across North America, Turtle Island. And I'm a settler on these lands, and I'm very grateful to be here. And I don't know if you noticed all the orange shirts yesterday, but I think we're still in the process of reconciling with what's happened over the last couple hundred years. Okay, so I don't know if you saw my caveat at the beginning. I'm not, I don't work on cancer specifically, but what I do work on is evolutionary rescue. And my hope is today to explain a little bit about what evolutionary rescue is and how it might be helpful in thinking about cancer drug resistance. So to start off, evolutionary rescue is an event. It's a population surviving when it would have went extinct without evolution. And I wasn't sure if there were chalkboards here, so I did this in my office late last night, just in case, but I should have known that fields would be covered in chalkboards. But the basic idea is, you have a population size on the y-axis and time on the x-axis. The solid line would be, what happened without evolution. The population goes extinct. However, with evolution, we may see a population adapt to this new environment and survive. So a little history on where the term comes from, evolutionary rescue. It was coined by Richard Gomulkiewicz and Bob Holt in a paper back in 1995. It's been a very important paper for the field and evolutionary theory in general. I'd also like to point out that Richard unfortunately passed away last year from cancer, and it was a very sad loss to the field as well as especially the evolutionary rescue community. But Richard and Bob were very interested in how a species niche or range limit might expand. So it's a very ecological context where this term comes from. So you can kind of maybe draw a picture something like this. Your species or population is able to grow in some portion of space or environment, and it's not able to grow in the rest of that space. And what happens is that individuals are constantly kind of being introduced to the area, perhaps outside the range edge, to the north of the range edge, or outside the niche generally. And the question is kind of, can these lineages that are arising in this new environment establish and expand the niche or the range of a species? So they wanted to understand this process, but this is a little bit complicated by this continual migration. So they first decided to consider the simpler case where you can think of n individuals moving out of the niche or the range, finding themselves in a new environment, and we cut off any future migration. So we're just dealing with the simpler case of n individuals dealing with a new environment. But this is, of course, totally equivalent to just the population in situ experiencing some abrupt environmental change. We can kind of ignore the kind of movement aspect of that. So this is, equivalent to a tumor population facing a new drug that the patient has taken. So evolutionary rescue, that was back in '95. This was coined. This has been 30 years of this field developing. There's quite a few papers, but I think I can kind of roughly summarize the field in two main axes. One is the environmental change, where on the one hand, like when we're thinking about drug resistance, we're thinking about an abrupt environmental change. The environment changes suddenly once. Or we can think on the other end of that spectrum, a very gradual environmental change. This is like, climate warming and climate change. So that's kind of one axis. The other axis is phenotype, where you can think of kind of a discrete trait, which is also quite common in drug resistance. You have resistant or not resistant, like we saw in the last talk. Or you can go to the other extreme of continuous, where it might be something like body size or your ability to, what temperature you prefer as an organism. So that phenotype becomes a continuous variable. And I think within these two axes, you can position kind of subfields of evolutionary rescue. I think drug resistance tends to be focused on these abrupt drug environments with discrete traits, because resistance is often coded by just a few genes, few loci. Conservation, on the other hand, tends to think about perhaps more gradual environmental changes, more complex phenotypes that are more continuous, but there is definitely some substantial overlap in this. I've probably made them look more separate than they really are. And this, I think this framework helps explain a little bit, at least, not everything for sure, the separation between drug resistance and evolutionary rescue for conservation. And this is the literature back in 2014 that Helen Alexander and others put together a network of evolutionary papers they consider to be about evolutionary rescue. And this is the field that I'm more familiar with down here, where we have the kind of population genetics, evolutionary theory, conservation, perhaps focused studies in green. And in blue, we have all the drug resistance literature. And that's like bacterial resistance and viral resistance and cancer. And you would know these citations up top better than I do. But these ones are kind of where I feel at home down here. But you see, there are some links. And the hope of, my hope in interacting at this workshop is to kind of strengthen the links between these blue and green dots here. So I'm going to tell you, was that listed in that? I'm going to tell you about this, this paper here, number 3, which is what I consider to be the kind of standard model for the abrupt environmental change, discrete trait version of evolutionary rescue, which is what I think is most applicable to drug resistance. So the standard model comes from this Orr and Uncliffe paper, 2008, in the American Naturalist. It's quite a straightforward setup. It has one locus that we're looking at with 2 alleles, big A and little a. We're dealing with just haploid selection. So you either have selection on the big A allele or selection on the little a allele. We don't have to worry about heterozygotes. And we're going to deal with it in discrete time, non-overlapping generations. The basic components of the model are that the wild type allele big A has a fitness of 1 minus R. So it's declining at rate R. Sorry, 1 minus R is less than 1. So we're declining at rate R. And the mutant allele, little a, has a fitness of the wild type multiplied by some benefit, 1 plus S. So it does a little bit better than the wild type allele. And when S and R are small enough, you can approximate this as 1 plus S minus R, where we assume that S is greater than R, so that this mutant allele is able to grow, has fitness greater than 1 in the new environment. And then we'll have forward mutation from the wild type to the mutant at rate mu. Okay, so the first thing you might want to understand is how many mutations arise during the wild type's decline. So the wild type dynamics are illustrated by the initial population size times its geometric decline, 1 minus R to the T. And then each generation, you're also getting a fraction of those mu that are mutant alleles. You sum that up over all the generations until this population will eventually go extinct, and you get the total number of mutations that arise during the wild type's decline. Then if we can calculate the probability that a new mutation establishes, which we'll call P, and in this population genetic literature, this is often approximated as 2 times the growth rate, which comes from branching process, approximation from the branching process. The probability of rescue from new mutations, which I'll denote DNM for de novo mutations, is given by this quantity here. And the basic idea is that, with probability 1 minus P, a new mutant doesn't establish. And so rescue is 1 minus the probability that no mutation establishes. So this 1 minus P to the total number of mutations is the probability of no mutation establishing. We just take 1 minus that. All we're cared about is in at least one mutation establishing. And when P is small, we can approximate this with this exponential. So this is kind of the classic form where we think of a kind of Poisson distribution of successful mutations, and this is the probability that that we have 0 of those. And that is, essentially the key result of the paper. It's not very, it's not that complicated. The other thing we can think about are not just the new mutations that arise during the wild type's decline, but mutations that existed prior to the environmental change, right? And so we can imagine we have just some number of those and little a times 0, and we calculate essentially the same thing. We replace the number of mutants that arise with the number of mutants that were already there, and we multiply them by their establishment probability. So this is the probability of evolutionary rescue by standing genetic variation. Now, the number of mutants is often something small and is probably quite liable to stochasticity. So if you need to deal with something at, say, mutation, selection, genetic drift balance, you may have to integrate over this distribution of what NA is to get a more accurate probability of rescue. But we won't need that. We won't need that for today. So we can put these two together. The probability of rescue can be written from either de novo mutation or standing genetic variation. Can be written like so. It's 1 minus the probability we don't have rescue by de novo, and we don't have it by standing genetic variation. So this is the fundamental, this is what did I call it? The conventional or the classic model of rescue in a abrupt change with a single discrete trait. But this has been, I'm giving you the simplest version. There have been a ton of extensions. This was 2008. So it's been quite a while of people elaborating on this. Things have been done like looking at diploidy in a fairly trivial case in that same paper, but also in more complex cases where the heterozygote can't grow, but the double mutant can, and the effect of mating system, et cetera, on that by Hildegard here in the crowd. People have worked on a single locus, but more than 2 alleles. In fact, an infinite number of alleles, each allele having a different fitness effect in this new environment. Hildegard has also worked on when you don't have just a single locus and 2 alleles, but you have 2 loci with 2 alleles, potentially with recombination, which is perhaps of less interest for the cancer crowd, but very relevant for many other systems. And people have worked on things where you need perhaps multiple mutations to accumulate in order to get resistance or adaptation to the new environment, which is one of the things I worked on a few years back. And the list goes on. People have worked on density dependence, where you get these ideas of competitive release. You get frequency dependent interactions between individuals. You have spatial structure, which could be quite relevant for the cancer world. You have species interactions, predation, host parasite, competition, etc. And there are a number of decent reviews on this. Some of them are getting a little dated now. So Hildegard and I and many others are working on a new review of evolutionary rescue theory at the moment. And when we did our literature search to figure out what we had to read and understand and synthesize, we found roughly 200 papers that were kind of under the term evolutionary rescue and mathematical model in the past 10 years. So I think this is, a rapidly accumulating literature, which is getting kind of more complex and sophisticated pretty quickly. Very few cancer papers showed up here, but we know that many exist, but just not using the same word. So to get back to cancer, one thing we can do here would just be to apply the theory as is. If drug resistance in a tumor arises by a single mutation at a single locus, then we can pretty much just apply this as is. The probability of treatment failure, in this case, is the probability of evolutionary rescue. Those are the same thing. What we're trying to do is minimize the probability of rescue. Or, of course, we could explore more complicated scenarios or hypotheses we have about drug regimen or genetic basis or what have you, with many of the extensions that have been developed in the last 10 years. Those 200 papers, plus the things that were done before that. So there's a lot more tools in the toolbox from rescue that can help understand drug resistance in cancer, I think. And what I'm going to do is to demonstrate an example of each of these in cancer drug resistance. So the first paper I'm going to talk about, neither of these papers am I a part of. I just was trying to find connections between rescue and cancer. And these are two very recent papers that I thought use evolutionary rescue in the title, which helped me find them, and I think are quite illustrative of the connection here. So the first example is two-strike therapy, which I think you can basically apply out of the box, the Ornunculus result that I just showed you. And the other case is resistance via aneuploids. And this requires a little bit more of the extension work, but not too much. Okay, so you all know more about this than I do, but the idea about two-strike therapy is that there's often resistance arising to the first drug that you give to a patient. So the obvious thing to do perhaps is to give a second drug, right? That this resistant population hopefully isn't resistant to. The question then becomes, when do you apply the second drug? I think for practical purposes, this happens when you see the tumor again, when the tumor has already gotten large. But there's also ideas in the literature that you should apply that, not when the tumor is grown up to some size again, but when it was at its minimum population size before relapse. But it's not clear exactly when to apply. So the model, sorry for the low quality there. The model consists of 4 different genotypes. There's a susceptible cell type. There's a cell type that's resistant to the first drug. There's a cell type that's resistant to the second drug. And there's a cell type that's resistant to both the first and the second drug. And these are the ones you really want to avoid. We're going to deal with just kind of one-way mutation. So the susceptibles mutate rate mu1 to the resistant ones, and to R2s with mutation rate mu2, and then these can mutate to become double mutants. The 3 resistant types have growth rates GR, and those growth rates are the same as the susceptible growth rate, but with a cost, like we heard in the last talk, some cost of resistance and metabolic cost. So it's these GRs slightly less than GS. And then depending on the environment, there'll be death rates added to this model. So in drug 1, the susceptibles and the R2s will get an extra death rate. In the second drug, the susceptibles and R1s will have an extra death rate due to that drug. And here's my drawing again. The idea here is that, the tumor starts some point in the past. The susceptible cells are growing. There's maybe some mutation selection balance of the resistant cells that are arising from the susceptibles. And then we start treatment at time 0. So at time 0, we apply the drug, and the susceptible cells decline exponentially. The resistant cells do 2 things. Those cells that are resistant to drug 1 can start to increase. They're now able to grow in this environment. And the cells that are resistant to drug 2 will remain at some low frequency due to mutation from the susceptibles and from the susceptibles. And now this is what we want to know. Where do we put this tau? When do we apply the second drug?
Quick question.
Yep.
Is there a reason why the resistance are not growing while it's susceptible?
Back here? Yeah, the
growth population or what?
So this is the tricky part about presenting someone else's work, but I think the rough idea, I think it's a little vague what's happening before the first drug is applied. Essentially, what you care about is the initial number of susceptible cells and the initial number of resistant types. So you can kind of ignore the past and just specify it, and which is what they do for most of the paper. You just give a number that seems reasonable for the initial number. But they also calculate a mutation selection balance number for these, which is some fraction of the susceptible population. So perhaps in that case, this should be tracking the increase of the susceptibles, but I didn't draw it accurately. Yeah. And so what they do is they treat this first drug dynamics deterministically. Okay, so we're going to treat this decline of the susceptibles and the growth of R1 and the low frequency of R2 deterministically, and then we're going to switch on the stochasticity and look at the probability of rescue at tau, and that's what we're trying to minimize. So where do we put tau to minimize it? Yeah, so I didn't draw that because what happens here once we hit the second drug, these individuals can now establish. So these cell lineages can establish, and the question is, will they, given this initial number of them? And so if you do get resistance, this will grow exponentially. If you don't get resistance, this will peter out. So this is kind of I probably shouldn't have drawn any lines here, but I think it's fairly safe to kind of think about the R1s and the S's as exponentially declining in this second drug. And the question is, do these R2s or mutations that are spawning out of these S's and R1s rescue the population? I'll try to make that clear with the equations that they use. So if you think about kind of the rescue from standing genetic variation, a little too high for me to point at, so I'll use this one. We have our same expression of 1 minus e to the negative times the establishment probability. And now we're trying to calculate the number of resistant mutations we have at the time that drug 2 is applied. And so the simpler term perhaps to think about is just this R2 at time tau. That's what I was just pointing out. I'll probably have to go back and forth here. So just how many R2s do we have at the end of this deterministic phase? That's one quantity that we want to minimize to reduce the probability of rescue. But there's also mutations that are arising during that deterministic phase from 0 to tau these are the double mutants that are arising. So the resistant type 1s are becoming double mutants at rate mu2, and the resistant type 2s are becoming double mutants at rate mu1. So these lineages here are double mutants that can establish even back in drug 1, right? Because they're resistant to drug 1. If they establish then, they'll just keep on growing because drug 2 doesn't bother them. So this is kind of double mutants that establish during the deterministic phase, and these are the type 2 resistant cells that establish after we add the second drug. Is that roughly clear? I guess the details don't matter too much, perhaps, but I think it's a pretty interesting model. So then the other way you can get resistance is via de novo mutation. And in this case, we have this exponential decline of the S and R1 cells in drug 2, and those are mutating at rate mu2 to either R2 cells from S or R12 cells from R1, and they'll establish with some probability P. So you can put the results together and calculate the probability of rescue, which will depend on tau through these integrals, as well as this initial number of R2s. And we need to find the tau that minimizes this probability of rescue. So I think this is a very nice application of evolutionary rescue theory that's pretty direct from the Orr-Unkel's result, with the added complication that instead of, basically the dynamics in the standing genetic variation are a little bit more complicated because we have this deterministic declines and increases, but it's essentially just a more complicated standing genetic variation, and we apply the same framework too. I won't go into all the intuition here, but they find that, as expected by previous theory, that the best time to switch from drug 1 to drug 2 is at very near the minimum population size that you reach with S declining and R increasing. So there's some minimum population size that's reached here, which is where it's the best time to apply the drug. With the caveat that it tends to be, at least under their parameter values, the best population size is to do a little bit, the best time is to do a little bit after the minimum, because that gives a little bit more time for these R2 cells to decline during drug 1. So you want to really make sure that any cells that are resistant to type 2 that have been sticking around before the drugs were applied, you really want to make sure you get rid of those with drug 1 before you apply drug 2. But you also don't want to let the double mutants get to high frequency in R1 or the single mutant R1s to get too high, because then they'll give you more de novo mutation via this term here. So there's a balance you're trying to find, and near the minimum but slightly after is the best. Okay, so the other study is about resistance by aneuploidy. And the idea here is that, again, you guys know the biology better than I do, but cancer cells have a high mutation rate to aneuploidy. We get kind of extra copies of chromosomes appearing. And this aneuploidy can actually sometimes be adaptive in drug. It can actually be protective. Yep.
Yeah, you have to tell me what aneuploidy is.
Aneuploidy is an extra copy of a chromosome. So you might have, during mitosis, you might by accident end up with 3 copies of chromosome 2 instead of 2. And sometimes having that extra copy of the chromosome means maybe you can increase the amount of protein you're producing or something, and that can change the phenotype, and that might be protective under a drug. And my understanding is that aneuploidy goes crazy in cancer lineages, and chromosome numbers go pretty wild. So this happens. So there's, those are the 2 main points. You get a high rate of mutation to aneuploidy, and it might have a benefit in drug. So there's a worry that aneuploidy will lead to drug resistance for these reasons. And the question becomes, by how much? How much does aneuploidy help a tumor survive a drug? In this case, the model has three types. There's a sensitive cell, the aneuploid cell, and the fully resistant mutation. The classic, thinking here is that sensitives become resistant at some mutation rate. Ignore their notation. I'm going to stick with mutation, the notation that I've been using. We're going to go sensitives become resistant at some mutation rate. Sensitives can also become aneuploidy at some aneuploid at some higher mutation rate, and then the aneuploids become resistant at the same rate the sensitives do. That's a a point mutation or something that happens at a given rate. Then these all have growth rates and death rates. There'll be some cost to resistance, but you can survive in the drug, etc. That part's pretty much the same. The idea then, like the benefit of aneuploidy, is that as the number of susceptible cells, sensitive cells, declines with the drug, you might get mutation to aneuploids. Then depending on the type of the effect of the aneuploid, you might just decline more slowly if it just gives you some tolerance. That might allow you to basically stop declining if it is near criticality, if this aneuploid is like near resistant, or it might even allow you to grow if the aneuploid confers some actual resistance. And then the resistant mutation is thought to be able to grow even faster in the presence of the drug. They also can. I think this is just an illustration of the growth rates. But of course, yeah, you can also get red going to green and having the resistant cells appear. And that's actually the kind of the key question is which pathway do you take and what's the rate of rescue via the two different pathways competing at the same time. So if we think about just the direct pathway from sensitives to resistant mutations, then this is exactly the result we saw from Orrin Unclis, that the probability of evolutionary rescue, I'm just going to deal with de novo mutations in this case, because that's what they do in the paper. You just have, well, that's mostly what they do in the paper. So the direct mutation rescue probability, you just have your mutation rate mu from sensitives to resistant types times the initial tumor size divided by the rate at which the tumor is declining, and then the probability of establishment of a resistant cell. And you can similarly calculate the probability of rescue via aneuploidy, which is a little bit more complicated. We have the mutation rate to aneuploids times the initial population divided by the decline rate. And then you have this X, which is a placeholder for something slightly more complicated, but has been derived a number of times. It's been derived in a model by Awasa back in 2004. Guillaume Martin and others have worked on this under the banner of evolutionary rescue back in 2013. And Guillaume and I have worked on this again in 2020 about how you have a rescue via multiple mutations. And so X can be approximated in three different cases. In the case where the aneuploids themselves are just resistant to the drug, then it's pretty straightforward. It's the analogous to direct mutation. You just replace PR by PA, the probability that an aneuploid cell establishes. And when aneuploids are not resistant, when they're declining, where we saw that, for in this case here, where you're just declining more slowly, then we need to multiply by the mutation rate from aneuploids to resistant types divided by the total number of, multiply by the total number of aneuploids we expect in a given lineage, which is 1 over R, times the probability of establishment of the resistant type. So it's kind of just applying this reasoning twice. You get a first mutation, then that lineage might have a second mutation. In between that, when aneuploids are nearly resistant, it's a little bit more complicated, but essentially from a two-type branching process, you just take an approximation in three different ways, and you can recover these three terms. And this can also be understood in some more heuristic ways, which is what the authors of the paper do as well to derive this. But there's three kind of scenarios. Yeah.
Does it assume that the population is steady state or it's growing?
So we're doing all of this under a branching process framework. So everything is just exponentially growing in this drug. So the wild types are exponentially declining, or the census is exponentially declining, and there's no interaction between any of these terms. There's no density dependence or anything like that. Does that make sense?
Yeah.
Yeah, and that's a classic assumption in not all of, but a lot of evolutionary rescue theory. Is the idea is basically, you start up where density dependence matters, and once the population starts declining, you can kind of ignore that competition density dependence term and treat things as branching processes for the most part. But there are exceptions, and people have incorporated competition as well. So the way the authors understand these results is that they say that the probability of rescue, which we might be able to write something like this, the initial tumor size times some per lineage probability of establishment, is roughly 1 when the initial tumor size is greater than 1 over the probability of establishment, which they call n star. So this is kind of n star is your kind of critical size. If the tumor is larger than n star, then rescue is quite likely. If the tumor is smaller than n star, then rescue is unlikely. And therefore, the conclusion is that if the n star via aneuploidy is less than the n star via direct mutation, then aneuploidy can cause treatment failure. So, if your tumor that you're looking at falls in between these two, then you can get rescue via aneuploidy, but when you'd be unlikely to get it via direct mutation. That will depend, of course, on the parameter values that we have in these expressions, and you can kind of work out when will aneuploidy be bad for treatment. And they have, I forget which cancer they're looking at, but they have nice parameter values estimated for a particular system where they can say something more precise and more practical. So this is perhaps a little short, but evolutionary rescue, I'm just going to summarize. Evolutionary rescue theory has grown quite quickly over the last 15 years. It is readily applied to drug resistance in cancer, like I think we're beginning to see now. There's been 4 or 5 papers in the last couple of years which kind of use the term evolutionary rescue and connect with the theory that's been developed there. And I think workshops like this and papers like these ones are helping create this crosstalk between the population genetics, ecology side of things and the cancer side of things. And I think that the part that I don't know about is all the cancer models that I think probably can help us understand rescue more generally. And I think that the general theory about evolutionary rescue can help understand cancer, as I think these papers demonstrate. With that, just thank everyone for listening, the organizers for inviting me, Hildegard for letting me bounce ideas off of her, and NSERC for funding.